In the quiet depths of the ocean, where sunlight fades to a distant memory and pressure builds with every meter of descent, a shadow moves through the water with a grace that seems almost impossible. It glides forward without effort, its body perfectly shaped by hundreds of millions of years of evolution, carrying within it an arsenal of abilities so extraordinary they would seem like science fiction if they weren't absolutely real. This is a shark, one of nature's most perfectly engineered predators, and the powers it possesses are nothing short of remarkable. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. Tonight, we're diving into the fascinating world of sharks, creatures that have survived 5 mass extinction events and continue to thrive in oceans around the globe. But these aren't just survivors. Sharks are holders of superpowers, abilities that push the boundaries of what we thought possible in the animal kingdom. They can detect electrical fields so faint they're measured in billionths of a volt. They can smell a single drop of blood diluted in an Olympic-sized swimming pool. They possess a sense that lets them feel the subtlest movements in the water around them. And they carry teeth that regenerate endlessly throughout their entire lives. Some can see in near total darkness, controlling light with mirror-like structures in their eyes. Others have skin that acts as both armor and engineering marvel, reducing drag as they slice through water. And perhaps, most astounding of all, certain shark species can live for centuries, with the oldest known individual swimming through waters that have changed beyond recognition since before the United States was founded. These aren't mythical creatures from ancient stories. These are real animals, swimming in our oceans right now, carrying within them solutions to challenges that human engineers and scientists are only beginning to understand. So, settle in, let your breathing slow, and join us as we explore the incredible superpowers of sharks. Beneath the surface of every ocean, invisible rivers of electricity flow through the water. Every living creature generates these fields, tiny pulses of bioelectricity created by the contractions of muscles, the firing of neurons, the basic processes of life itself. For most animals, these electrical signatures pass unnoticed, nothing more than background noise in a busy world. But sharks possess a sense that makes them aware of this hidden dimension of reality, a superpower that lets them perceive what remains invisible to almost every other creature on Earth. This is electroreception, and it's powered by one of the most sensitive biological sensors ever discovered. The story of how scientists came to understand this remarkable ability is itself a tale of curiosity and patient observation. For centuries, fishermen and naturalists noticed peculiar dimples on the heads and snouts of sharks, tiny pores that seemed to serve no obvious purpose. In 1678, an Italian physician named Stefano Lorenzini described these structures in detail, documenting the way they connected to jelly-filled canals beneath the shark's skin. But their function remained a mystery for nearly 300 years. It wasn't until the 1960s that researchers finally began to unlock the secret of what came to be known as the ampullae of Lorenzini. The breakthrough came through careful experimentation. When scientists placed electrodes near these mysterious pores and introduced weak electrical fields into the water, they observed something remarkable. The sharks responded. Their behavior changed. Nerve recordings showed that signals were racing from these organs to the shark's brain. Slowly, piece by piece, the picture became clear. These weren't just pores. They were the openings to one of nature's most sophisticated sensory systems. Each ampulla of Lorenzini consists of a small pore at the surface of the skin, leading down through a canal filled with a clear, jelly-like substance. This gel is no ordinary material. It possesses one of the highest proton conductivities of any biological substance, allowing it to transmit electrical signals with remarkable efficiency. The canal, which can be several centimeters long in larger sharks, terminates in a small chamber lined with specialized sensory cells. These cells are exquisitely sensitive to changes in electrical potential, responding to differences between the voltage at the skin surface and the voltage deep within the tissue. The sensitivity of this system defies belief. Sharks can detect electrical fields as weak as 5 nanovolts per centimeter. To put this in perspective, imagine taking a 1.5 volt AA battery and connecting its terminals with wires that span the entire width of the Atlantic Ocean. A shark could detect the resulting electrical field. This makes them more sensitive to electrical stimuli than any other animal known to science. Even the sophisticated electroreceptive capabilities of electric fish, which generate and sense their own electrical fields, don't match the pure sensitivity of a shark's ampullae. But what does this actually mean for the shark as it moves through its world? The applications are both numerous and profound. When a fish swims, its muscles contract and relax, generating small electrical fields with each movement. When it breathes, electrical signals pulse through its gills. Even when it lies perfectly still, buried in sand and hoping to avoid detection, its heart continues to beat, and with each beat, a tiny electrical signal radiates outward into the surrounding water. A shark swimming overhead can detect these signals. The buried prey might be invisible to the shark's eyes, impossible to smell if the current is flowing the wrong direction, completely silent, but it cannot hide its electrical signature. This ability transforms the way sharks hunt. Picture a hammerhead shark sweeping its distinctive head back and forth above the seafloor like a metal detector scanning a beach. That's essentially what it's doing, except instead of searching for coins and jewelry, it's searching for the electrical signatures of hidden prey. The wide spacing of its ampullae across that hammer-shaped head gives it enhanced directional information, allowing it to pinpoint the location of buried animals with remarkable precision. When it detects the telltale electrical field of a stingray concealed beneath the sand, it doesn't need to see or smell its prey. It knows exactly where to strike. The ampullae aren't distributed evenly across a shark's body. They cluster most densely around the head, particularly on the underside of the snout and around the mouth. This makes perfect sense when you consider the final moments of a hunting sequence. As a shark closes in on its prey, preparing to bite, its eyes often roll back into its head for protection, a behavior observed in many species. In that crucial moment, the shark is essentially blind, yet it must still aim its attack with precision. The ampullae of Lorenzini step in to fill this gap, providing the shark with detailed electrical information about its target's exact location, even when visual information is unavailable. The number of these sensory organs varies dramatically between species. Some sharks have a few hundred ampullae, while others possess several thousand. The sawfish, a relative of sharks, holds the record with more ampullary pores than any other cartilaginous fish, suggesting that this group has evolved to be specialists in electroreception. The differences in ampullae count and distribution likely reflect each species' particular ecological niche and hunting strategy. But electroreception isn't solely about finding food. Sharks also appear to use these organs for navigation, tapping into one of Earth's most fundamental features, its magnetic field. Our planet generates a vast magnetic field, created by the churning of molten iron in its core. This field extends far out into space, protecting us from harmful solar radiation, and it also penetrates through the oceans. Ocean currents, being composed of salt water, conduct electricity, and as these currents flow through Earth's magnetic field, they generate weak electrical fields of their own. Sharks can detect these fields. Research suggests that sharks may create mental maps of the electrical signatures associated with different ocean currents and magnetic variations. A great white shark, migrating thousands of kilometers across open ocean, far from any visible landmarks, may be navigating by following these invisible electrical highways. They're reading the very structure of the planet's magnetic field, translated into electrical information their brains can process. It's a form of perception almost alien to us, humans living in a world dominated by light and sound. Yet for sharks, it's as natural as breathing. The jelly filling the ampullary canals serves multiple functions beyond simply conducting electricity. Scientists initially thought this substance might also help sharks sense temperature gradients, acting as a kind of thermoelectric semiconductor that could convert heat differences into electrical signals. While that particular hypothesis has largely been disproven, researchers in 2023 predicted that sharks might be able to detect temperature differences as small as 1/1000th of a degree using their ampullae. Whether this proves true remains to be seen, but it hints at layers of complexity we're still working to understand. What's particularly fascinating is how this electrical sense integrates with the shark's other abilities. A shark doesn't hunt with just one sense. It uses them all in sequence, each taking over as it becomes most useful. From great distances, hearing and smell dominate. As the shark gets closer, vision becomes important. Then the lateral line system kicks in, detecting water movements. And finally, in those last crucial moments, the ampullae of Lorenzini provide the precise targeting information needed to complete the hunt. It's a symphony of senses, each playing its part at exactly the right time. The discovery of electroreception in sharks fundamentally changed our understanding of how these animals interact with their environment. It revealed a hidden layer of reality that we, as humans, simply cannot access without sophisticated electronic equipment. And perhaps, most remarkably, sharks have been using this ability for hundreds of millions of years. Long before the first mammals walked on land. Long before the first birds took flight, since the ancient seas of the Paleozoic era. Evolution has had an enormous amount of time to refine this system, and the result is nothing short of a superpower. While sharks possess this extraordinary electrical sense, they also carry with them one of the most celebrated and feared abilities in the animal kingdom, the power of smell. The ocean is full of scents, chemicals dissolved in water that tell stories about food, danger, potential mates, and the presence of competitors. For sharks, smell isn't just one sense among many. It's often their primary tool for making sense of their world, the sense they rely on most heavily when searching for their next meal across vast stretches of seemingly empty ocean. Scientists sometimes call sharks swimming noses, and this description isn't far from the truth. A large portion of a shark's brain is dedicated to processing olfactory information. In some species, the olfactory bulbs, the brain structures that analyze smell, can account for up to 2/3 of the brain's total weight. This massive neural investment reflects just how crucial the sense of smell is to a shark's survival. The physical structure of a shark's olfactory system is elegantly designed for its purpose. Unlike humans, whose nostrils serve double duty for both breathing and smelling, sharks keep these functions completely separate. They breathe through their gills, and their nares, the equivalent of nostrils, are dedicated exclusively to detecting scents. Each nare has 2 openings, one for water to flow in and one for it to flow out. As a shark swims, water continuously streams through these channels, passing over a series of thin, leaf-like structures called olfactory lamellae. These lamellae dramatically increase the surface area available for detecting scent molecules. Imagine the way a book's pages multiply the available reading surface compared to a single flat sheet. The olfactory lamellae work on the same principle. Each one is lined with sensory cells, and when a scent molecule in the water makes contact with the right receptor, it triggers a nerve impulse that races to the brain. The more lamellae a shark has, the more opportunities it has to intercept and identify scent molecules flowing past. The sensitivity of this system is legendary. Laboratory experiments have demonstrated that sharks can detect certain chemicals at concentrations as low as 1 part per 10 billion. Some research suggests they can respond to specific amino acids, the building blocks of proteins, at even more dilute concentrations, perhaps as sparse as 1 molecule in 10 to the 15th molecules of water. To grasp the magnitude of this sensitivity, imagine trying to detect a single drop of blood in an Olympic-sized swimming pool. That's the scale we're talking about. In some tests, sharks have been documented detecting fish extracts at these almost impossible dilutions. But extraordinary sensitivity is only part of the story. A shark also needs to know where a scent is coming from, and this presents a significant challenge. When a source of scent releases chemicals into the ocean, those molecules don't spread out in a neat, uniform sphere. They form an irregular plume, twisted and stretched by currents into long, turbulent corridors that can span hundreds of meters. Within this corridor, concentrations fluctuate wildly. The distribution is chaotic, patchy, unpredictable. Traditional wisdom held that sharks might track scents by comparing concentrations between their 2 nares, turning toward whichever nostril detected the stronger signal. But the physics simply doesn't support this idea. The distance between a shark's 2 nostrils, even in large species, is too small for there to be a meaningful concentration gradient at the distances involved. So, how do they do it? The answer lies in timing. Research has shown that sharks can detect incredibly small delays in when a scent reaches one nostril versus the other. We're talking about differences measured in fractions of a second, gaps so brief that they would be imperceptible to us. When a shark encounters a plume of scent at an angle, the odor molecules reach the front nostril slightly before they reach the rear one. By comparing these arrival times, the shark's brain can calculate which direction the scent is coming from. As the shark swims, it moves its head from side to side in a characteristic scanning pattern. This isn't random motion. With each sweep, the shark samples the water on both sides, gathering information about the distribution of scent in space. If the signal strengthens to the left, the shark adjusts course to the left. If it weakens, the shark might swing its head the other way, searching for the stronger part of the plume. This is a dynamic process, constantly updating, constantly correcting, guiding the shark toward the source through three-dimensional space. The distances over which sharks can detect and track scents depend on many factors: the nature of the chemical being released, the strength of the source, water temperature, salinity, and especially the behavior of ocean currents. Under ideal conditions, when a large quantity of scent material enters the water and a strong current carries it directly toward a shark, detection can occur from hundreds of meters away. The often cited figure of a quarter mile isn't unrealistic for a substantial blood scent in good conditions, though it's worth noting that this represents a best-case scenario. More typically, sharks probably detect food-related scents from somewhat shorter ranges, perhaps a few dozen to 100 meters in most situations. What's particularly interesting is how different species have evolved variations on this basic system to suit their particular lifestyles. Hammerhead sharks, with their dramatically widened heads, space their nostrils much farther apart than most other sharks. Research on bonnethead sharks, small hammerheads with relatively modest head expansions, has confirmed that they can indeed detect and respond to concentration gradients between their widely separated nares. The greater spacing gives them enhanced directional information, potentially allowing them to orient to scent sources with more precision than their narrower-headed cousins. This may help explain the evolution of that distinctive hammer shape, a feature that's puzzled scientists for generations. Not all sharks are equally obsessed with their sense of smell. Species that hunt by ambush, waiting motionlessly for prey to swim within striking distance, tend to have less developed olfactory systems. They rely more heavily on vision and their ability to detect vibrations in the water. But for the cruising predators, the ones that roam open water, searching for their next meal, smell is absolutely critical. It's their long-range detection system, the sense that tells them when and where to invest their energy in pursuit. The chemicals sharks find most attractive aren't necessarily blood, despite the popular image. Research has shown that sharks respond strongly to certain amino acids and to the oils and fluids released from fish tissues. They're tuned to detect the chemical signatures of injury, of struggling prey, of potential food. While they certainly can detect blood, they're generally more interested in the broader chemical profile of a wounded animal. This makes sense from an evolutionary perspective. A shark that can identify the scent of easy prey, animals that are already injured or distressed, has a significant advantage over competitors who waste energy chasing healthy, difficult-to-catch targets. Humans sometimes worry about attracting sharks with bleeding wounds while swimming in the ocean, but the reality is more nuanced than popular culture suggests. Yes, sharks can detect blood in the water, but they're detecting many other things too, and blood from a small cut on a human finger is unlikely to be particularly attractive to a shark that has evolved to hunt fish, seals, or squid. The concentration matters, the context matters, and most importantly, sharks aren't mindless eating machines that attack anything that smells vaguely like food. They're selective predators that have learned over millions of years to identify the scent signatures of their preferred prey. The olfactory sense also plays a role beyond hunting. Sharks use scent to identify potential mates, to detect pheromones that signal reproductive readiness. They may use it for navigation as well, recognizing chemical landmarks in the ocean, distinctive scent profiles that mark familiar territories or migration routes. The smell of home waters might be as recognizing to a shark as the sight of familiar streets is to us. What scientists are still working to fully understand is how the shark's brain processes and interprets all this olfactory information. How does it distinguish between the scent of prey and the scent of a competitor? How does it know the difference between a struggling fish and a dead one that's already too decayed to be good food? These are questions that require more research, more careful observation of how sharks behave in response to different chemical cues. But what's already clear is that this sense of smell is far more sophisticated, far more central to the shark's existence than most people realize. The ocean is never still. It ripples and surges, driven by wind, tide, the passage of boats, and the movements of countless animals swimming through its depths. Every action creates a response, a pattern of vibrations and pressure waves that radiate outward through the water. Most of these disturbances are tiny, barely perceptible, even with sophisticated instruments. But to a shark, they're as clear as footsteps on a creaking floor. This is the domain of the lateral line system, a sensory apparatus that allows sharks to feel the ocean's movements and detect the presence of nearby objects, even in complete darkness. If you were to look closely at a shark's skin, you would see faint lines running along its body, typically starting near the head and extending back toward the tail. These lines mark the location of canals, narrow tunnels just beneath the skin's surface filled with fluid. Opening to the water through tiny pores, these canals contain structures called neuromasts, clusters of sensory cells that respond to movement. Each neuromast consists of hair cells, not hair in the sense of what grows on your head, but microscopic cellular projections called cilia that extend into a gelatinous cupula. When water moves, even slightly, it displaces this cupula, bending the cilia. And when the cilia bend, they trigger nerve impulses that inform the brain about the water's movement. The lateral line system is ancient, shared by essentially all fish, and present even in the aquatic larval stages of amphibians. It evolved early in vertebrate history, hundreds of millions of years ago, and it's been refined over that vast span of time into a remarkably sophisticated sense. For sharks, it serves as a kind of distant touch, a way of feeling things without making direct contact. It's particularly useful for detecting low-frequency vibrations, the kind of rumbling, rhythmic movements created by swimming prey. Think about how a fish swims. Its body flexes side to side, its fins and tail pushing against the water, creating vortices and pressure waves with each stroke. These patterns are distinctive. Different species produce different movement signatures. A healthy fish swimming normally creates one pattern. An injured fish, struggling to maintain its position, creates another. To a shark's lateral line, these differences are obvious. From meters away, a shark can sense that something is moving, and it can gather information about that object's size, speed, and behavior without seeing it or smelling it. The lateral line is especially important in murky water, where visibility is limited, and in darkness, when vision provides little useful information. It's also crucial in the final moments of an attack. As we mentioned earlier, many sharks roll their eyes back when striking, protecting these delicate organs from the thrashing of captured prey. During these moments, the lateral line helps guide the bite, providing real-time feedback about the prey's exact position and movements. The distribution of neuromasts isn't uniform across a shark's body. They cluster more densely around the head, and many sharks have a particularly rich concentration on their snouts. This makes sense, given the snout's role in first contact with prey. Having enhanced mechanoreception in this region allows the shark to make fine adjustments to its strike, to respond to last-second movements by its target. The lateral line on the body, meanwhile, provides information about water flow and pressure changes along the shark's sides and tail, helping it maintain balance and optimize its swimming efficiency. Interestingly, scientists believe that neuromasts come in two main varieties. Superficial neuromasts sit on the skin surface or in shallow pits, exposed directly to water flow. These tend to be sensitive to lower frequency vibrations and water velocity. Canal neuromasts, enclosed within the lateral line canals, are more protected and respond better to higher frequency signals and acceleration. This dual system gives sharks the ability to process a wider range of mechanical information, capturing both the slow, powerful movements of large prey and the rapid, delicate vibrations that might indicate something smaller or more distant. The lateral line doesn't work in isolation. It's intimately connected with the shark's hearing system, and in fact, the two are sometimes referred to together as the acoustico-lateralis system because they share similar sensory cells and evolutionary origins. Both respond to vibrations, but they operate in different frequency ranges. Hearing picks up higher frequency sounds that travel long distances through water, while the lateral line specializes in lower frequencies and closer range detection. Together, they give the shark a comprehensive picture of the acoustic and mechanical landscape of its environment. Research has shown that sharks can use their lateral line to detect objects in their path, even stationary ones. As water flows around an obstacle, it creates characteristic patterns of pressure and movement. A shark swimming toward a rock or a piece of kelp can sense these patterns and adjust its course to avoid collision. This is part of how sharks navigate through complex environments like coral reefs, where visual cues might be limited and obstacles are everywhere. The lateral line provides a kind of hydrodynamic image of nearby space. The precision of the lateral line system is hard to quantify, but behavioral studies give us hints. Sharks can detect the vibrations from struggling fish from considerable distances, probably several body lengths away at least. They can distinguish between different types of movement, between prey and non-prey, between something worth investigating and something to ignore. They can sense the direction a disturbance is coming from and adjust their swimming to intercept it. All of this happens without conscious thought, processed by neural circuits honed by evolution to extract meaningful information from the constant background noise of ocean movement. One fascinating aspect of the lateral line is how it remains effective even as the shark itself is moving. When you're swimming forward, you create your own bow wave, your own turbulence. How does the shark's brain separate self-generated noise from signals that actually matter? The answer involves sophisticated neural processing. The system includes inhibitory pathways that can dampen responses to expected patterns of stimulation, effectively filtering out the shark's own movements to make external signals more apparent. It's comparable to how your brain filters out the sound of your own footsteps when you're walking, so you can better hear other sounds around you. As with many of the shark's senses, the lateral line's capabilities vary between species based on their ecological needs. Bottom-dwelling sharks that hunt in sand or mud, like many species of rays, which are close relatives of sharks, rely heavily on their lateral line to detect buried prey. Pelagic sharks that hunt in open water might use it more for schooling behavior and long-range detection of distant movements. Each species has tuned its system to the particular challenges of its lifestyle. The integration of all these senses, electroreception, olfaction, the lateral line, creates a perceptual world vastly different from our own. Sharks don't just see the ocean, they feel it, smell it, sense its electrical dimensions. They exist in a rich sensory landscape that we can barely imagine, surrounded by information flowing in from every direction. And as we'll continue to discover, this is just the beginning of their superpowers. In the realm of biological weaponry, few things are as iconic or as formidable as a shark's teeth. Rows upon rows of serrated triangles, some as large as your hand, designed to grip, tear, and crush with devastating efficiency. But what truly makes these teeth remarkable isn't just their size or sharpness. It's the fact that they never stop coming. Throughout a shark's entire life, from the moment it's born until the day it dies, it continuously produces new teeth, replacing worn or lost ones with fresh, sharp replacements, in an endless cycle that represents one of nature's most effective solutions to a common problem. Consider for a moment the challenge that teeth present for most animals. They're exposed to tremendous forces. They chip, they crack, they wear down from use. They decay. For mammals like us, teeth are precious resources that must be protected because we only get two sets, our baby teeth and our adult teeth. And if we lose those adult teeth, they're gone forever. Our bodies cannot make more. This limitation shapes our entire approach to dental care, and it's why tooth loss can be so traumatic. Not just cosmetically, but functionally. Without teeth, eating becomes difficult, nutrition suffers, and survival is threatened. Sharks have solved this problem in the most straightforward way imaginable. They never stop making teeth. It's continuous production, a biological assembly line that operates from birth to death without pause. A single shark may produce tens of thousands of teeth over its lifetime, perhaps even more. The exact number varies by species and individual lifespan, but estimates suggest that some sharks may generate 30,000 or more teeth in their lifetime. When you consider that many shark species live for decades, and Greenland sharks can live for centuries, the cumulative tooth production becomes almost incomprehensible. The mechanism behind this perpetual tooth factory lies in specialized structures within the shark's jaw. Early in development, before the first teeth even appear, the embryonic shark forms a region called the dental lamina, a band of tissue that contains stem cells capable of giving rise to teeth. In most mammals, the dental lamina produces teeth a couple of times and then disappears. In sharks, it never goes away. It remains active throughout the animal's entire life, constantly generating new teeth. The teeth themselves grow in rows, typically anywhere from 3 to 7 rows, depending on the species, though some sharks have more. In a great white shark, you might see 5 rows of teeth at any given time, with several hundred teeth present in the mouth simultaneously. But here's the fascinating part. Only the frontmost row is actively functional. The teeth behind it are in various stages of development, waiting their turn. They lie flat against the jaw tissue, gradually maturing and hardening. As teeth in the front row wear down or break off, which happens frequently given the forces involved in hunting and feeding, the row behind them rotates forward to take their place. This replacement happens on a conveyor belt principle. The whole row moves forward, not just individual teeth. It's a coordinated process driven by the continuous growth of new teeth from the dental lamina combined with the shedding of old teeth at the jaw margins. The rate of replacement varies considerably between species and even within species based on factors like age, diet, and environmental conditions. Some species replace teeth as quickly as every week or 2. Others take several months. For a large predatory shark that routinely bites into tough prey, tooth loss is a frequent occurrence, so rapid replacement is essential. Scientists studying this process have identified the genetic pathways that control it. The genes involved are ancient, conserved across hundreds of millions of years of evolution. They're members of signaling pathways with names like hedgehog, OWNT, BMP, and FGF, molecular systems that play crucial roles in development across all vertebrates. In mammals, these same genes are active during our 2 phases of tooth development. The difference is that in sharks, they remain switched on permanently, redeploying again and again to create each successive generation of teeth. Recent research has revealed that the continuous production of teeth in sharks depends on a population of stem cells that reside in the dental lamina. These stem cells maintain their capacity to divide and differentiate into tooth-forming cells throughout the shark's life. They're the ultimate source, the reservoir from which all those thousands of teeth ultimately derive. Understanding how sharks maintain these stem cells without exhausting them, how they keep them active for decades or centuries without depleting their regenerative potential, is a question that fascinates researchers. We humans also had these stem cells once, during our development, but we lost them early in life. The tissue that housed them degrades and disappears. If we could learn how to reactivate similar stem cells in adult humans, we might someday be able to grow replacement teeth naturally, instead of relying on artificial implants. The structure of shark teeth varies dramatically between species, reflecting different hunting strategies and dietary preferences. Great white sharks have large, triangular, serrated teeth, perfect for cutting through the flesh and blubber of marine mammals like seals. Tiger sharks have curved, heavily serrated teeth that can saw through turtle shells and crush bones. Mako sharks have long, thin, pointed teeth designed for gripping slippery fish that might otherwise escape. Some species of sharks have flat, pavement-like teeth for crushing shellfish and crustaceans. Each tooth shape is an adaptation, fine-tuned over millions of years to the specific foods that shark eats. But regardless of shape, all shark teeth share certain fundamental properties. They're not made of bone, they're composed of dentine, a hard tissue similar to what forms the core of human teeth, covered with a layer of extremely hard enameloid. This enameloid is what gives shark teeth their incredible durability and sharpness. It's so hard that fossilized shark teeth, which can be millions of years old, still retain their sharp edges. In fact, The majority of the shark fossil record consists of teeth because the rest of the shark's skeleton is made of cartilage, which doesn't fossilize as readily as bone. Teeth, being mineralized and hard, persist long after the soft tissues and cartilaginous skeleton have decayed away. The teeth aren't rooted into the jaw the way mammalian teeth are. Instead, they're embedded in the gum tissue, held in place by connective tissue that's strong but flexible. This loose attachment serves an important function. When a shark bites into something tough, like a large fish or the body of a seal, individual teeth might take extreme forces. If they were rigidly attached, they might fracture or damage the jawbone. The flexible attachment allows teeth to give a little, to bend and twist with the forces, reducing the risk of catastrophic damage. And if a tooth does break or get dislodged, it's no great loss. Another one will move forward to replace it soon enough. Interestingly, some shark species can actually replace their entire set of teeth simultaneously, rather than on a row-by-row basis. The cookie cutter shark, for instance, sheds its entire lower jaw dentition at once, swallowing the teeth perhaps for the calcium and other nutrients they contain, while a fresh set rotates into place. This is relatively unusual behavior, but it demonstrates the flexibility of the system. The rate at which sharks lose teeth varies depending on what they're biting into. A shark that regularly feeds on hard-shelled prey, or that hunts large, struggling animals, will lose teeth more frequently than one with a softer diet. Studies have tracked tooth loss in captive sharks and found that some individuals can lose dozens of teeth per year. In the wild, where hunting is more unpredictable and violent encounters more common, loss rates are probably even higher. The continuous replacement system means this isn't a problem. It's built into the shark's biology, expected, accounted for. This endless regeneration of teeth represents a profound difference between sharks and most other vertebrates. It's a reminder that evolution can arrive at radically different solutions to the same problems depending on the circumstances and the starting materials available. Mammals, including humans, are descended from ancestors that evolved on land where conserving resources like calcium was important. Having teeth that lasted a long time made sense in that context. Sharks, meanwhile, remained in the ocean, where calcium is abundantly available in dissolved form. The evolutionary pressure to make teeth last forever wasn't there. Instead, there was pressure to always have sharp, effective teeth available, and the solution was continuous replacement. The study of shark tooth regeneration isn't just academically interesting. It has potential practical applications. If researchers can understand the signals that keep shark dental stem cells active, they might be able to develop therapies to regenerate teeth in humans. This is a long way off, and there are enormous challenges involved in translating insights from sharks to people given the hundreds of millions of years of evolutionary divergence between us. But the fundamental biology, the core genetic toolkit, is surprisingly well conserved. The same genes that build shark teeth also built your teeth when you were developing in the womb. Learning how sharks keep those genes active might someday help us do the same. For now, though, the shark's arsenal remains uniquely its own, a perpetual source of weaponry that ensures no matter how many teeth it loses in battle, there are always more waiting in reserve. Run your hand along a shark's skin and you'll immediately notice something unusual. In one direction, it feels relatively smooth. In the opposite direction, it's like sandpaper, rough enough to scrape your skin if you're not careful. This distinctive texture comes from thousands of tiny structures called dermal denticles, and while they might look like scales, they're actually more similar to teeth. In fact, denticles and teeth share a common evolutionary origin, and they're made from the same materials, a core of dentine covered with a hard layer of enameloid. But these aren't just protective covering. The dermal denticles serve multiple functions, and one of the most remarkable is their role in improving a shark's swimming performance. Scientists have long suspected that shark skin might reduce drag during locomotion, and decades of research have confirmed and refined this understanding, revealing that these tiny structures are marvels of natural engineering that human designers are still trying to fully replicate. Each dermal denticle is a tiny three-dimensional structure. Its exact shape varies between shark species and even between different regions of the same shark's body. But many fast-swimming sharks have denticles with longitudinal ridges running from front to back. These ridges are typically spaced just tens of micrometers apart, far too small to see clearly with the naked eye, but visible under a microscope. Collectively, they create a textured surface somewhat similar to the riblets that engineers sometimes add to aircraft or boat hulls to reduce drag. The physics of how this works is complex, but the basic principle involves managing the boundary layer, the thin region of water immediately adjacent to the shark's skin, where flow patterns are particularly important. As water flows over a surface, it tends to form turbulent eddies and vortices that increase drag. The denticle's ridges appear to suppress some of this turbulence, keeping the flow more laminar, more organized. This reduces the friction between the shark and the water, allowing it to swim faster for the same energy expenditure, or swim at the same speed with less effort. Laboratory experiments have measured the benefits. Researchers have built robotic flapping foils, devices that can swim through water in controlled ways, and covered them with real shark skin, or with synthetic materials designed to mimic denticle patterns. When they compare the swimming speed of these foils with and without denticles, they find measurable improvements. In some experiments, shark skin has been shown to increase swimming speed by over 12% compared to smooth surfaces. That's a substantial advantage. In the competitive world of marine predation, where capturing prey or escaping predators might come down to small differences in speed or endurance, a 12% improvement could mean the difference between eating and starving, between survival and death. Importantly, this benefit seems to require the flexibility of real skin. When researchers tested rigid plates covered with shark skin, the advantage largely disappeared. It's only when the skin is attached to a flexible, membrane-like surface that can deform naturally during swimming that the denticles provide their full drag-reducing effect. This suggests that the interaction between the denticles and the underlying flexible tissue is important, that the system works as an integrated whole rather than the denticles acting in isolation. The denticles may also enhance thrust generation, not just reduce drag. Computer simulations and flow visualization studies have shown that denticles can promote enhanced leading edge suction on fins and body surfaces. This additional suction contributes to forward thrust, effectively helping to pull the shark through the water. So, the denticles might be doing double duty, reducing the forces that slow the shark down, while simultaneously increasing the forces that drive it forward. It's an elegant solution, the kind of optimization that only evolution, working across millions of generations, can achieve. Different shark species have evolved different denticle shapes suited to their particular lifestyles. Fast-swimming species like the shortfin mako, which can reach speeds exceeding 30 kilometers per hour, have denticles with precisely spaced ridges that fall within the optimal range, predicted by fluid dynamics theory. Slower swimming species that spend much of their time near the bottom have denticles with different shapes, sometimes lacking ridges altogether. These denticles might be optimized more for protection or anti-fouling than for hydrodynamic performance, reflecting the different priorities of a less active lifestyle. The protective function of denticles shouldn't be underestimated. The enameloid covering makes them about as hard as granite and as strong as steel, providing the shark with an effective suit of armor that doesn't sacrifice mobility. This armor protects against multiple threats. Large predators, including other sharks, might attack, and the denticles make it harder for their teeth to gain purchase. Smaller parasites trying to attach to the shark's skin must contend with the rough, closely packed denticles that offer few secure footholds. Some research suggests that the denticles may also resist biofouling, the accumulation of algae, barnacles, and other organisms that commonly encrust surfaces submerged in seawater. A clean skin is important for maintaining optimal swimming efficiency. The denticles also contribute to silent swimming. Large bony fish with their overlapping scales create turbulence and noise as water flows over their bodies. Sharks, with their hydrodynamically optimized denticles, swim much more quietly. If you were to place a hydrophone in the water near a cruising shark, you'd hear very little swimming sound. This stealth is valuable for a predator. Prey species often have keen hearing and can detect the sounds of an approaching threat. A silent predator has a better chance of getting close before being noticed. Engineers have been inspired by shark skin denticles for decades, attempting to create artificial surfaces that provide similar benefits. In the 1990s, a company developed swimsuits with fabric designed to mimic shark skin texture, claiming they would reduce drag and help swimmers go faster. These suits became controversial when they were associated with numerous world records, leading to debates about whether they provided an unfair advantage. Eventually, suits with these features were banned from competition. More recently, engineers have explored using shark skin-inspired surfaces on boat hulls, aircraft, and wind turbine blades, anywhere drag reduction might save energy or improve performance. But creating effective biomimetic shark skin has proven challenging. Early attempts often increased drag rather than reducing it. Getting the three-dimensional geometry right, the precise shape and spacing of the ridges, the material properties, the interaction with the underlying surface, all of these factors matter. Nature spent millions of years evolving shark denticles. Human engineers are working to condense that process into a few decades, and it's not easy. Still, progress is being made, and as our understanding deepens and our manufacturing capabilities improve, we may yet succeed in creating artificial surfaces that truly capture the benefits of shark skin. One particularly intriguing avenue of research involves the idea that sharks might actively control their denticles. Some species, like the shortfin mako, have denticles that appear to be able to bristle, changing their angle relative to the skin surface. If sharks can indeed adjust their denticles dynamically, perhaps raising them during rapid maneuvers to alter flow patterns and reduce pressure drag. That would represent yet another layer of sophistication in an already remarkable system. This remains an active area of investigation, with researchers trying to determine whether and how sharks might exercise such control. What's clear is that dermal denticles are far more than just a protective covering. They're an active component of the shark's locomotor system, a piece of evolutionary engineering that improves swimming efficiency in ways we're still working to fully understand. They're also a reminder that nature often arrives at elegant solutions to complex problems, solutions that human technology is still struggling to match. Most fish, when they want to control their vertical position in the water column, rely on a swim bladder, a gas-filled organ that provides buoyancy much like a balloon might lift a weight. By adjusting the amount of gas in the bladder, a fish can make itself more or less buoyant, allowing it to hover at a particular depth without sinking or floating to the surface. It's an efficient system, widely used throughout the fish world. But sharks don't have swim bladders. They never evolved them. Instead, they've developed a completely different solution to the buoyancy problem, one that's elegant, effective, and reveals yet another of their superpowers. The key to a shark's buoyancy lies in its liver. If you were to dissect a shark, one of the first things you'd notice is the size of this organ. In some species, the liver can account for a quarter or even a third of the animal's total body weight. It's enormous, often divided into two large lobes that extend through much of the shark's abdominal cavity. And it's not just big, it's full of oil. This oil is much less dense than seawater, and it's this property that provides the shark with lift, helping to offset the weight of its denser tissues, like muscle and cartilage. The primary component of shark liver oil is a substance called squalene. Squalene is a hydrocarbon, part of the biochemical pathway that leads to cholesterol synthesis in most organisms. It's present in small amounts in many living things, including humans, where it's a component of our skin's natural oils. But in sharks, particularly deep-sea sharks, squalene is produced in vast quantities and stored in the liver in concentrations that can exceed 90% of the oil's total composition. A large shark might have hundreds of kilograms of squalene-rich oil in its liver. Squalene has a density significantly lower than water, about 870 grams per litre, compared to seawater's roughly 1,027 grams per litre at typical ocean temperatures. This density difference creates buoyant force. When the shark is submerged, the oil-filled liver displaces water that weighs more than the oil itself, and this generates upward lift. It's the same principle that makes a boat float. Displace water that weighs more than your vessel, and you experience a net upward force. What makes squalene particularly well suited for this role is its behavior under pressure. As a shark dives deeper, the pressure increases dramatically. Every 10 meters of depth adds roughly 1 atmosphere of pressure. At depths of several hundred meters, the pressures are immense. Most substances compress significantly under these conditions, becoming denser as their molecules are forced closer together. If squalene compressed too much, it would lose its buoyancy benefit. But research has shown that squalene's compressibility is remarkably similar to that of seawater itself. As you go deeper, both compress at nearly the same rate, so the density difference between them remains almost constant. This means that a shark's liver provides stable buoyancy across a wide range of depths, from the surface down to the deepest waters where sharks are found. This is quite different from how a swim bladder works. A gas-filled bladder compresses dramatically as pressure increases, losing buoyancy unless the fish actively adds more gas to it, a process that requires energy and time. For a shark, making rapid vertical movements, diving and ascending as it hunts or migrates, an oil-filled liver is a more practical solution. The buoyancy is relatively consistent, requiring less active management. But there are trade-offs. Even with a large, oily liver, most sharks are still slightly negatively buoyant. They sink if they stop swimming. This is particularly true for large, fast-swimming species like great whites, blue sharks, and makos. These sharks must swim continuously to generate hydrodynamic lift from their fins, using their pectoral fins somewhat like airplane wings to keep themselves from sinking. This isn't necessarily a disadvantage. Continuous swimming keeps water flowing over the gills, providing oxygen. It also allows these sharks to cover large distances as they search for food or migrate between feeding and breeding grounds. Deep-sea sharks have taken the oil-filled liver strategy to an extreme. Species that live at depths of several hundred to several thousand meters, where pressures are enormous and food is scarce, have evolved livers that can make up an even larger proportion of their body mass than their shallow water cousins. Some deep-sea sharks are very close to neutrally buoyant, able to drift slowly in the water column with minimal effort. This is crucial for conserving energy in an environment where the next meal might be weeks or months away. The Greenland shark, which we'll discuss in more detail shortly, is a classic example. Its massive, oil-rich liver allows it to move through the deep, cold waters of the Arctic with minimal energy expenditure. The composition of liver oil varies between shark species. While squalene is dominant in many deep-sea sharks, particularly those in the order squaliforms, other groups use different lipid compounds. Sharks in the order carchainiforms, which includes many common coastal species, store more triacylglycerols, or tags, which are denser than squalene. Chimeras, close relatives of sharks, use another class of compounds called diacylglycerol ethers. Each chemical has slightly different physical properties, and the choice of which to use likely reflects the particular depth range, temperature regime, and lifestyle of each species. Sharks may have some ability to adjust their buoyancy by altering the composition of their liver oil. Research on dogfish sharks suggested that they might respond to changes in buoyancy, such as having weights attached to them in experimental settings, by shifting the balance of lipids in their livers, increasing the proportion of lighter compounds at the expense of heavier ones. This adjustment takes time, probably weeks or months, so it's not a rapid response mechanism, but it could help sharks fine-tune their buoyancy over seasonal migrations, or as they grow and their body composition changes. The reliance on liver oil for buoyancy has had consequences beyond just how sharks swim. It's made sharks targets for humans. Shark liver oil, and squalene in particular, has been harvested for commercial use for decades. Initially, it was sought for vitamin A extraction, particularly during World War II when traditional sources were unavailable. After synthetic vitamin A became available, the demand shifted to other uses. Squalene is valued in cosmetics for its moisturizing properties. It's used in some vaccines as an adjuvant to enhance immune response. These commercial uses have driven fishing pressure on shark populations, particularly on deep-sea species with large, oily livers. Many of these deep-sea sharks are slow-growing and late-maturing, making them especially vulnerable to overexploitation. Conservation concerns have prompted efforts to find alternative sources of squalene, such as extracting it from plant oils, though shark-derived squalene remains cheaper and more abundant, perpetuating the demand. From a purely biological perspective, though, the oil-filled liver is a masterstroke of evolutionary engineering. It solves the buoyancy problem without the complexity of a swim bladder. It provides stable lift across a range of depths and temperatures. It does so using materials that the shark's body produces as part of normal metabolism, and it allows sharks to occupy ecological niches, particularly in the deep sea, where other predators struggle to survive. It's yet another example of sharks taking a different path from most other fish and arriving at a solution that works brilliantly for their particular way of life. In the dark depths of the ocean, far below where sunlight can penetrate, vision might seem useless. And yet, many deep-sea sharks have large, well-developed eyes, suggesting that even in these perpetually dark environments, sight remains important. The secret lies in bioluminescence, the light produced by various marine organisms, and in the shark's own remarkable adaptations for seeing in near-total darkness. Among these adaptations, one of the most extraordinary is a structure called the tapetum lucidum, a biological mirror that fundamentally changes how a shark's eye works. The basic structure of a shark's eye is similar to that of other vertebrates, including humans. There's a cornea that helps focus incoming light, a lens that refines that focus, and a retina at the back of the eye where photoreceptor cells detect light and convert it into neural signals. These photoreceptors come in two main types: rods, which are sensitive to light intensity and work well in low light conditions, and cones, which respond to different wavelengths and enable color vision. Most sharks have a high proportion of rods relative to cones, reflecting their need to see in dim environments. But what truly sets a shark's eye apart is the tapetum lucidum. This structure sits behind the retina, a layer of cells containing guanine crystals, arranged in a precise, mirror-like configuration. These crystals are highly reflective. When light passes through the retina without being absorbed by a photoreceptor, instead of just disappearing into the tissues behind the eye, it hits the tapetum and bounces back through the retina a second time. This gives the photoreceptors another chance to detect it. Essentially, it's a way of amplifying the available light, making the eye roughly twice as efficient at capturing photons. If you've ever seen a cat's eyes glowing in the dark when a light shines on them, you're seeing the tapetum lucidum at work. Sharks have the same feature, and in some species, it's even more effective than in cats. Measurements suggest that a shark's tapetum can be up to twice as efficient at reflecting light as a cat's tapetum. This enhanced efficiency allows sharks to see in conditions that would leave most other animals effectively blind. Estimates suggest that sharks can see about 10 times better than humans in dim light, a truly remarkable advantage for an animal that often hunts at dawn, dusk, or in the murky depths where light is scarce. The tapetum isn't a static structure. In many shark species, it can be adjusted. During bright light conditions, such as when a shark approaches the surface on a sunny day, specialized pigment-containing cells can migrate to cover the reflective crystals, effectively blocking them off and preventing the tapetum from functioning. This protects the retina from being overwhelmed by too much light. The process is reversible. When light levels drop, the pigment cells retract, uncovering the tapetum and restoring the eye's enhanced sensitivity. This adaptation happens relatively slowly compared to the rapid dilation and constriction of the pupil, taking minutes to hours rather than seconds. But it allows the shark to function across a much wider range of lighting conditions than it could otherwise. The pupil itself is another interesting feature. In many sharks, the pupil can change shape dramatically. In low light, it opens into a large, round aperture, maximizing the amount of light entering the eye. In bright light, it contracts into a narrow slit, similar to what you see in a cat's eye. This ability to adjust pupil size and shape allows the shark to regulate light intake on a much faster timescale than the tapetum's pigment migration, providing immediate protection when moving from dark to bright environments. Shark eyes are positioned laterally on the head, one on each side, which gives them a very wide field of view. A shark can see almost all around itself, with only small blind spots directly in front of the snout and directly behind the head. This panoramic vision is valuable for detecting threats and opportunities from any direction. However, it comes at the cost of reduced binocular vision. The regions where the visual fields of the two eyes overlap are small, which means sharks have limited depth perception compared to animals with forward-facing eyes. But this is less of a problem than you might think. Sharks compensate with their other senses, using their lateral line to detect nearby objects and their electroreceptors for precision targeting in the final moments of an attack. While sharks can certainly see, they don't rely on vision alone, and for many species, it's not even their primary sense. Smell and electroreception often take precedence. This is particularly true in murky water, where visibility is limited. But in clear water, especially for species that hunt visually, sight is critically important. Great white sharks, for example, are known to use vision extensively. They often hunt by spotting prey silhouetted against the bright surface when looking up from below. That distinctive attack from beneath, the massive surge upward that can even lift the shark partially out of the water, is typically a visually guided behavior. The question of whether sharks see in color has been debated for years. For a long time, it was thought that sharks were completely colorblind, seeing only in shades of gray. More recent research has challenged this view. Scientists have found that some shark species do possess cone cells in their retinas, suggesting at least some capacity for color vision. However, most sharks appear to have only one type of cone cell, unlike humans who have three types that allow us to distinguish red, green, and blue light. With only one cone type, sharks' color vision would be limited. They might be able to distinguish brightness and perhaps detect some color information, but they wouldn't see the rich palette of colors that we do. What sharks are exceptionally good at is detecting contrast, particularly between light and dark. This is crucial for spotting prey against varied backgrounds. A seal swimming near the surface appears as a dark shape against the brighter water above. A fish moving over a sandy bottom creates contrast between its body and the pale substrate. These differences in brightness are what a shark's eye is optimized to detect. The high proportion of rod cells, the tapetum lucidum, the ability to adjust to different light levels, all of these features support contrast detection. Motion detection is another area where shark vision excels. Rod cells respond not just to light intensity, but also to changes in that intensity over time. Something moving creates a changing pattern of light and shadow, and the shark's retina is very good at picking up on these changes. This is why remaining still can sometimes help you avoid a shark's attention. Though obviously, that's not a strategy you'd want to rely on if a shark is already actively hunting you. The shark's brain is wired to notice movement, to prioritize it as potentially important information that might indicate prey or threat. Different shark species have evolved different visual capabilities suited to their environments. Deep-sea sharks, as mentioned, often have larger eyes relative to their body size, an adaptation for capturing every available photon in the nearly lightless depths. Sharks living in murky coastal waters might have less acute vision and rely more heavily on their other senses. Reef sharks in clear tropical waters have eyes that are well suited for the bright, colourful environments they inhabit, though their colour perception is still probably limited compared to the fish they hunt. As with so many aspects of shark biology, vision doesn't work in isolation. It's part of an integrated sensory system. In the sequence of a hunt, different senses dominate at different ranges. Far away, hearing and smell are most important. As the shark closes to within a few dozen meters, vision becomes relevant. Closer still, the lateral line provides information about movements and vibrations. And in those final moments, electroreception takes over, guiding the strike when the eyes have rolled back for protection. Each sense has its role, and the shark's brain seamlessly integrates information from all of them to create a coherent picture of its environment. The study of shark vision continues to reveal new details and complexities. Researchers are using increasingly sophisticated techniques to measure visual acuity, contrast sensitivity, and color discrimination. They're examining how different species vary in their visual capabilities and how those variations relate to their ecology and behavior. And as our understanding grows, so does our appreciation for how sharks perceive their world, a world that looks and feels very different from the one we experience, but is no less rich in information and meaning. There are animals on this planet that have witnessed the rise and fall of empires, that were alive when Shakespeare was writing his plays, that were swimming through Arctic waters while the first European explorers were mapping unknown coastlines. These animals are Greenland sharks, and they represent, perhaps, the most astonishing superpower in the entire animal kingdom: the ability to live for centuries, persisting through timescales that make even the longest human lifespan seem fleeting. With potential lifespans reaching 400 years or more, the Greenland shark is the longest-lived vertebrate known to science. For many years, scientists suspected that Greenland sharks lived exceptionally long lives, but they had no way to prove it. The usual methods for aging fish involve counting growth bands in hard tissues, like fin spines or vertebrae, much like counting rings in a tree trunk. Each band represents a year of growth, but Greenland sharks, like all sharks, have skeletons made of cartilage rather than bone. Their vertebrae are too soft to form distinct annual growth bands. Without a reliable aging method, researchers could only guess based on indirect evidence. They knew that Greenland sharks grew extremely slowly, adding less than 1 centimeter to their length each year. They knew that some individuals reached sizes of 5 meters or more. Simple mathematics suggested that achieving such large sizes at such slow growth rates would require many decades, perhaps even centuries. But this was speculation, not proof. The breakthrough came through an ingenious application of radiocarbon dating. In the 1950s and 60s, atmospheric nuclear weapons testing released large amounts of carbon-14, a radioactive isotope of carbon, into the atmosphere. This carbon-14 eventually made its way into the oceans and was incorporated into the tissues of marine organisms. The peak occurred in the mid-1960s, after which levels began to decline as the carbon-14 dispersed and decayed. This historical spike in carbon-14 provides a time marker. By measuring the amount of carbon-14 in tissues that formed during an animal's development, researchers can estimate when that development occurred. For Greenland sharks, the key tissue turned out to be the eye lens. Unlike most body tissues that are constantly being rebuilt and replaced, the proteins in the core of the eye lens are laid down before birth and then remain unchanged throughout the animal's life. They're like fossils, preserved within the living shark. When researchers extracted these lens cores from dead sharks, mostly animals caught accidentally in fishing nets, and measured their carbon-14 content, they could estimate the birth year of each shark. A study published in 2016 analysed 28 female Greenland sharks using this technique. The results were stunning. The largest shark in the study, measuring just over 5 meters in length, was estimated to be between 272 and 512 years old. Even at the lower end of that range, 272 years, this would make it the longest-lived vertebrate ever documented, surpassing the previous record holder, a bowhead whale that lived to at least 211 years. The uncertainty in the estimate is large because radiocarbon dating has limitations. But even accounting for that uncertainty, the conclusion is inescapable. Greenland sharks live for centuries. The implications of this longevity are profound. These sharks reach sexual maturity only after about 150 years. A female Greenland shark isn't ready to reproduce until she's older than most nations, older than the United States as a country, older than the Industrial Revolution. The gestation period is estimated to be 8 to 18 years, among the longest for any vertebrate. Everything about the Greenland shark's life history happens in slow motion, stretched out over timescales that challenge our human understanding of what it means to be an animal. Why do they live so long? The answer probably lies in their environment and their metabolism. Greenland sharks inhabit the cold, deep waters of the North Atlantic and Arctic Oceans, environments where temperatures hover just above freezing. Cold slows down biochemical reactions. Metabolism in cold-adapted animals tends to be much slower than in warm-adapted ones. The Greenland shark's metabolic rate is exceptionally low. It swims at an average speed of only 34 centimeters per second, one of the slowest swimming speeds recorded for any fish of its size. Its tail beats at an extremely low frequency. Everything about it suggests an animal that moves through life in the slowest of slow lanes, conserving energy at every turn. This slow metabolism may contribute to longevity by reducing the accumulation of cellular damage. One of the major theories of aging suggests that oxidative stress, damage caused by reactive oxygen species produced during normal metabolism, gradually degrades cellular structures and DNA over time. If your metabolism is slower, you produce less oxidative damage per unit time, and your cells might remain functional for longer. Additionally, slow growth and delayed maturity are often associated with long lifespans across many animal species. It's as if the pace of living correlates with the total duration of life. Recent genomic studies have begun to shed light on the genetic underpinnings of Greenland shark longevity. In 2024, researchers successfully sequenced the Greenland shark's genome, finding it to be extraordinarily large, roughly twice the size of the human genome and the largest shark genome sequenced to date. Within this genome, they identified multiple copies of genes involved in DNA repair and inflammation control. These duplicated genes might provide the shark with enhanced ability to fix damaged DNA and manage inflammation, both of which are important for maintaining health over long time spans. Other genes associated with longevity in model organisms, such as those involved in cellular stress responses, also appeared to show interesting patterns in the Greenland shark. The longevity of Greenland sharks raises fascinating questions about aging itself. Aging is often thought of as an inevitable process, a progressive decline that all organisms must undergo. But the existence of animals that can live for centuries challenges that notion. If a Greenland shark can maintain functional organs, swim, hunt, and reproduce after 400 years, what's fundamentally different about its biology compared to ours? What cellular and molecular mechanisms allow it to avoid or repair the damage that would kill most other animals in a fraction of that time? Understanding these mechanisms could have implications not just for basic biology, but potentially for human health and aging research. There are practical challenges to studying these sharks. They live in remote, inhospitable environments. They spend much of their time at depths that are difficult to reach and observe. They're rarely caught, and when they are, it's usually as accidental bycatch. This makes it hard to gather large sample sizes or conduct detailed behavioral studies. What we know comes from piecing together information from scattered observations and the occasional specimen that researchers can examine. But each piece of information adds to the picture. One intriguing aspect of Greenland shark biology is their apparent resistance to the diseases and degenerative conditions that typically affect long-lived animals. You might expect that after centuries, a shark's tissues would show signs of accumulated damage, cellular senescence, tumors, and other age-related pathologies. Yet, the limited evidence available suggests they remain remarkably healthy even at advanced ages. The oldest sharks examined don't appear to be riddled with cancer or struggling with organ failure. They seem to just keep going, maintaining their physiological function far longer than seems reasonable. This resistance to age-related disease is itself a superpower, one that medical researchers are very interested in understanding. The conservation implications of such extreme longevity are sobering. An animal that takes a century and a half to reach reproductive age is extraordinarily vulnerable to overfishing. If you remove a significant number of mature individuals from a population, it will take more than a human lifetime for that population to recover, if it can recover at all. Greenland sharks are currently listed as vulnerable by conservation organizations, and fishing pressure, particularly the historical hunting for their liver oil, has likely had significant impacts on their populations. Protecting them isn't just about saving individual sharks. It's about safeguarding entire generations that span centuries, lineages that connect the deep past with the far future. Imagine, for a moment, a Greenland shark born in 1620. It would have been a juvenile when Galileo was observing the moons of Jupiter, establishing the foundations of modern astronomy. It would have reached adulthood around the time of the American Revolution. It would have been in its prime during the Victorian era, might have been swimming through icy waters while the Titanic sank, while 2 world wars reshaped human civilization, while humanity first ventured into space. And if that shark is still alive today, it continues to swim in the same cold, dark waters, largely unchanged by the passage of centuries. A living link to a world that has transformed beyond recognition everywhere except in the depths where it dwells. This perspective is humbling. It reminds us that our human timescales, the decades we live, and the centuries we remember through history are not universal. Other forms of life experience time differently. For a Greenland shark, the events that seem monumental to us, the wars, the technological revolutions, the rise and fall of nations, all of that is just a fleeting moment in a lifespan that stretches across multiple human generations. And yet, despite living so differently, in a realm so removed from our own, they face threats from human activities: fishing, pollution, climate change. The coldest parts of the ocean are warming. How this will affect these ancient sharks, whether they can adapt on their glacial timescales to changes happening in mere decades, remains an open and troubling question. The Greenland shark reminds us that the superpowers of sharks aren't just about hunting or survival in a short-term sense. Some of their abilities operate on timescales that dwarf our own, stretching the very definition of what an individual life can encompass. And in doing so, they challenge us to think differently about longevity, about aging, about what's possible in the realm of biology. And so, we reach the end of our journey through the superpowers of sharks. We've explored abilities that seem almost magical, senses that detect invisible forces, biological systems that regenerate endlessly, bodies optimized by hundreds of millions of years of evolution to be among the most effective predators the planet has ever produced. But perhaps, more than anything, what we've learned is that sharks are far more than the frightening monsters of popular imagination. They're sophisticated, complex animals that have solved the challenges of life in the ocean in remarkable ways. They can feel the electrical whispers of hidden prey through sensors more sensitive than any human-made instrument. They can smell a single molecule of blood diluted in an Olympic swimming pool and track its source across hundreds of meters of open water. They can sense the subtlest pressure waves and vibrations through a sensory system that turns their entire body into a detector for movement and disturbance. They carry teeth that regenerate continuously throughout lives that can span decades, or, in one extraordinary case, centuries. Their skin isn't just covering, but engineered armor that reduces drag and increases swimming efficiency. Their livers act as biological buoyancy tanks, filled with oils that let them navigate to the depths. Their eyes can see in near darkness, amplifying faint light with biological mirrors. And some of them live longer than any other vertebrate on Earth, persisting through timescales that make human history seem brief. These aren't just interesting facts. They're solutions to fundamental problems faced by all living things. How to find food, how to avoid becoming food, how to move efficiently, how to maintain your body over time. Sharks have answered these questions in ways that continue to inspire scientists and engineers. From biomimetic swimsuits to dental regeneration research, from underwater robotics to longevity studies, sharks are teaching us lessons about design, function, and possibility. But these extraordinary creatures face uncertain futures. Shark populations worldwide are under pressure from overfishing, habitat loss, and climate change. Many species are declining, some critically. And because sharks are slow to reproduce, with many species taking years or decades to reach maturity, populations that decline can take generations to recover. The loss of sharks from ocean ecosystems would have cascading effects, disrupting food webs and altering the balance of marine communities in ways we're only beginning to understand. As apex predators, they play crucial roles in maintaining healthy oceans. Understanding sharks, truly appreciating their biology and their place in the natural world, is a step toward protecting them. They're not mindless killing machines. They're highly evolved animals with complex sensory systems, sophisticated behaviors, and ecological roles that have been refined over an evolutionary timescale measured in hundreds of millions of years. They deserve our respect, our wonder, and our efforts to ensure they continue to swim through the world's oceans for millions of years to come. If you've enjoyed learning about these incredible superpowers, if these facts have sparked your curiosity or helped you drift towards sleep while thinking about the wonders of the natural world, please consider subscribing to Bedtime Biology. Leave a comment with your thoughts or suggestions for future topics. Share this video with others who might appreciate a calm, detailed exploration of science. Each view, each subscription, helps us continue exploring the quiet marvels that surround us, the phenomena that inspire wonder when we take the time to look closely. Thank you for joining us tonight. May your dreams be filled with the gentle movement of ancient predators gliding through dark waters, perfectly adapted, endlessly fascinating, carrying within them secrets we're only beginning to unravel. Sleep well.